A very good afternoon to one and all present here. Honorable Vice Chancellor Sir, Professor Prateek Sharma, our esteemed guests, Professor Jyotsna Batra and Dr. Rupert Ecker, Head of Department Biotechnology, Professor Yasha Hasija, Convener of the event, Dr. Navneeta Bharadwaj, all faculty members, staff, and students, we welcome you to the second lecture series in biotechnology organized by the Department of Biotechnology, Delhi Technological University. I request all the dignitaries on the dais to please proceed for the lamp lighting. I request Honorable Vice Chancellor Sir to welcome our esteemed guests, Professor Jyotsna Batra and Dr. Rupert Ecker, with a sapling. Now I request Head of Department Biotechnology, Professor Yasha Hasija, to deliver the welcome address.
Good afternoon, everyone. Honorable Vice Chancellor Sir, Professor Prateek Sharma, our distinguished speakers, Dr. Jyotsna Batra and Dr. Ecker, Professor Praveer Kumar, Dr. Navneeta Bharadwaj, the convener of the lecture series, all the faculty members and dear students. It is with great pride and pleasure that I welcome you all to the second session of our lecture series in biotechnology. This initiative, envisioned by our Honorable Vice Chancellor Sir as a platform to bridge academic learning with real-world applications in biotechnology, has already witnessed an encouraging beginning with enthusiastic participation, insightful discussions, and a shared passion for scientific discovery. At the very outset, I'd like to extend my heartfelt gratitude to our Honorable Vice Chancellor Sir, Professor Prateek Sharma, whose visionary leadership and unwavering support have been instrumental in fostering a culture of academic excellence and research innovation at our university. Under his dynamic guidance, we continue to push boundaries, providing our students and faculty with opportunities to learn from leading experts in the field. Sir, it is with your encouragement and commitment to knowledge-driven initiatives that make events like this possible. Today, we are honored to welcome two eminent speakers who have made remarkable contributions to science and innovation. Dr. Jyotsna Batra, professor at Queensland University of Technology, is a renowned researcher in genomics and personalized health. With expertise in biochemistry and biotechnology, she has secured over $10 million in research funding and received prestigious accolades reflecting her significant impact on biomedical sciences and her dedication to advancing STEM education. On a personal note, I have the privilege of knowing Dr. Jyotsna since my PhD days at CSIR IGIB, where I witnessed her relentless work ethics and unwavering commitment to scientific excellence. I've always admired her dedication, her tireless pursuit of knowledge, and her ability to overcome challenges with grace and perseverance. I am confident that as you listen to her today, you'll be truly inspired by her success story and her passion for transforming scientific research into impactful real-world applications. Dr. Rupert Ecker, CEO of Tissue Gnostics, GmbH, and adjunct professor at QUT, is a pioneering expert in biomedical imaging and diagnostics. His work in automated tissue analysis and personalized medicine has revolutionized the field, making cutting-edge healthcare solutions more accessible and efficient. We are privileged to have such esteemed speakers with us today, and I'm thankful to both of them to have kindly accepted our invitation and be with us here today despite their packed schedule. I'm certain that their insights will inspire, challenge and encourage all of us to think beyond the conventional and explore new frontiers in biotechnology. Thank you.
Now, I request the Honorable Vice Chancellor, sir, to say a few words.
So good morning, all of you. So Dr. Rupert and Professor Jyotsna Batra, my distinguished colleagues on the panel, on the dais and in the audience, students, it's a real pleasure to welcome both of you. And in fact, I would like to compliment the Department of Biotechnology and the entire team for, first of all, being the first in the institution to initiate this lecture series and coming up with the calendar of the entire year-long lectures, which in fact we had conceived together to actually create a sort of a platform for the students as well as the faculty members, as I mentioned in the inaugural lecture, to actually give everyone an opportunity to be benefited from the experience of people who have distinguished themselves in their research and industrial application of biotechnology. And usually, at the cost of repetition, I would say, usually what happens is that when you want to, venture into new area or applications or any research, the normal course is that you visit the library, refer to research articles, or take your own initiative to use, say for example, YouTube or any other means through which you get to know. And in this case, the effort is entirely your own, and that again depends on your mood and other related factors, which at times becomes very difficult in your daily routines. Having people like the speakers of the day who are here, and the lecture series like this is this only, that when we invite such people, through their decades of experience, the information is presented in a capsule form, which again has a huge density of information, which otherwise becomes very difficult for anybody, teachers and students included, to gather in such a short time. And it also basically inspires and motivates students and faculty members again to know much more about a certain discipline or an area of application or research activity which is being undertaken by the researchers, the speakers who come and share their experiences. And one doesn't know where one thought, one idea basically becomes a topic of your research in future, or motivates you to undertake, several other initiatives in your life. Apart from that, in general, it also helps to add value to your existing knowledge, which otherwise becomes very difficult. Usually, conventionally, what happens is that we are always engaged in our routine activities of, attending classes, doing our assignments, going to the lab, which everybody in the institution or the country does. And so the difference comes when you bring a new dimension to that normal course. And this happens when we expose the students and faculty members alike to people who have been, working in a particular area for decades. And when they come and present through their talk, during that talk, one gets an opportunity to, enrich ourselves through their knowledge in a very limited short time, which actually is quite beneficial, and that is the purpose of having these lecture series. And again, I would like to really compliment the Department of Biotechnology of very swiftly putting the idea into action, which even though I would be politically incorrect to say, the other departments are struggling to even get their act in order. So this is how, the departments, people, researchers distinguish themselves in the execution, because the earlier you do, the better it is for everyone, and it gives you a sense of satisfaction. And this distinguishes a good department from an average department. So all my compliments to the entire team for taking this initiative. And I would take this opportunity to thank Professor Jyotsna and Dr. Rupert for very kindly agreeing to our request and share their experiences with the students and the faculty members. Thank you so very much. God bless you all.
I request the Honorable Vice Chancellor, Sir, to felicitate our esteemed guest, Professor Jyotsna Batra and Dr. Rupert Ecker. Now I would like to introduce our first speaker, Dr. Rupert Ecker. Sir graduated in cell biology from the University of Vienna. He has more than 25 years of experience in microscopy and image analysis. In addition, he has been trained in software development. Sir has been co-founder of the Tissue Gnostics Group in Austria, Romania, USA, Asia Pacific, and Australia. He is the Chief Executive Officer of Tissue Gnostic Global. Since August 2021, Sir has held an honorary appointment as an adjunct professor at the Faculty of Health, School of Biomedical Sciences, Queensland University of Technology, where he works on artificial intelligence-based decision support systems for next-generation histopathology, tissue cytometry. Please welcome, Sir.
Good afternoon, ladies and gentlemen, dear colleagues. Before I really start with my presentation and show you what I have done in my lifetime, more or less, I would like to start by and tell you that I obviously have not always been CEO and obviously also not the adjunct professor. I started like you as a student of cell biology, and I embarked on an adventurous journey from science to business, back into science. In fact, my company, which I'm a co-founder and co-owner of, is based on my PhD thesis. So during my PhD, I have made an invention which brings the idea of flow cytometry to the slide, and this is called tissue image cytometry. And it all started, well, a long time ago, in the year 1994, when I started my master's at the Medical University of Vienna Department of Urology. And one of my first jobs as a young student was to manually delineate nuclei and cytoplasm of cells, cultured cells, also on images that were scanned on a microscope. And I did this for weeks, from early morning to late evening, until I got the tendinitis from holding the mouse stiff the whole day. And I went to my boss and said, look, we are sending robots to Mars, which autonomously navigate on the surface of another planet. But we here in medical research sit like Stone Age people and draw circles. Stone Age people didn't have a computer, obviously, but all I did was drawing circles, telling the computer, this is a nucleus, this is a cytoplasm, this is a nucleus, this is a cytoplasm. And my boss looked at me and said, change it. So this was the start of a development which led to my PhD title years later, and ultimately also to the foundation of the company Tissue Gnostics. So I believe the message for you is, you are also embarking on a potentially very adventurous journey by studying here at this esteemed university. And this will open doors for you, which people who do not go through this education as you do will ever be locked. So really believe in yourself and work hard, and then many, many things will become possible. So I'm obviously wearing also two hats. The one as CEO and CSO, as a Chief Scientific Officer of Tissue Gnostics, and the other one is adjunct professor at Queensland University of Technology. And what I would like to show you is what my company has developed. I'm also the head of R&D, as a research and development. And the technology is called tissue image cytometry. So who of you is familiar with flow cytometry? Is this a term you know? Yeah? So flow cytometry has been available for maybe 50 years. It quantifies antibody-mediated staining on cells in solution. And if you look on a literature database like PubMed and look for as a search for flow cytometry or FACS, F-A-C-S, fluorescence activated cell sorter, you will find thousands of publications which over the decades which have used flow cytometry as tool of research and discovery. And what I wanted to do, and what I succeeded to do, in fact, is to bring the idea of flow cytometry that you can quantify antibody-mediated staining on individual cells from cell suspension to the tissue section and the microscopic slide. And this is what tissue image cytometry is about. We see here an example of a colon, and in brown, a very famous marker called Ki67. It's a proliferation marker, so that shows that these cells are about to divide. The algorithms we have in place allow to automatically segment, so identify the individual nuclei here. And you see with the in red contour are the Ki67 positive cells, in green are the Ki67 negative. And what you get is this type of data output, a so-called scattergram, or people in flow cytometry call it a dot plot, where each dot here represents a single cell in the image. We can also do what is nowadays called spatial biology by analyzing individual cells in spatial context. So which subpopulation of cells is related with which other? So where are they in the tissue? How far are they away from each other? The company was founded in the city of Vienna in Austria, Europe. And this is still in this beautiful building where our corporate headquarters is located. Nowadays we have a global network of subsidiaries and offices, and also distributors. Like here in India, we work through through distributors. And so Tissue-Gnostics is really a global entity dedicated to scientific excellence and the success in research by our clients. And I'm proud to say that as of today, we have more than 3,200 reference papers published by our clients and users around the world from more than 60 countries on all continents. Being a cell biologist myself, I've always been closely related to academic collaboration. And over the now 22 years which I'm running my company, I've had literally dozens of collaborative projects. What I would like to mention here are the most prestigious one. These are so-called Marie Curie Innovative Training Networks. This is a project from the European Union where they give money, a lot of money. Each of these projects that you see here receives anywhere between 3 and 6 million euro, where several universities from different countries in Europe come together with industry partners to work on specific topics and train students, early stage researchers. So these projects dealt with here digital pathology. This project was headed by the Department of Pathology at the University of Nottingham in UK. INSEM was the network of cell migration studies headed by the Technical University of Aachen in Germany. Alcatraz, the translational oncology network, headed by the University of Cambridge in UK. ALK is the anaplastic lymphoma kinase, a molecule very important in leukemias and lymphomas. Then we had the calcium sensing receptor network by Medical University of Vienna, and Helical, the big data network for clinical benefit with Trinity College Dublin. These 5 projects have already been finished, but ongoing are those 2. SSBB, Stop Spread Bad Bugs, focuses on the increasing problem of multi-drug resistance in bacteria, and this is run in collaboration, or led in fact by the Erasmus Medical Center in Rotterdam in the Netherlands, and Eradicate, the network on cancer stem cells and eradication of cancer relapse through the retinoic acid receptor and the vitamin D receptor at Medical University of Vienna. And you see there are different questions, but the common denominator in all those projects is that this research consortia had a need to have automated microscopy and image cytometry at the highest quality level, and this is what TissueGnostics offers. Our product line called TissueFax, so that the name of the technology is tissue image cytometry, the name of the products is TissueFax. Comes in different configurations. You might recognize that the microscope is in fact a Carl Zeiss microscope. So TissueGnostics is OEM, Original Equipment Manufacturing Partner of Carl Zeiss Germany. It has here typically a scanning stage for 8 slides, 2 cameras, a color camera for transmission microscopy, a monochrome camera for fluorescence microscopy. We also have configurations with spinning disc confocals, so you can do whole slide confocal imaging systems with slide loaders for 120 slides, batch scanning, or inverted system where you can also scan cell cultures and do live cell imaging in maybe even at a culture chamber. The software solutions we have developed, and this is really what TissueGnostics made TissueGnostics. They are called HistoQuest, TissueQuest, StrataQuest, and StrataQuest is what I call our flagship, which allows to run machine learning and deep learning algorithm, but also classical image cytometry. The initial output or intermediary result of these scanners is what is called a virtual slide. So you put your tissue section, which is on a glass slide, on the microscope, and it's automatically scanned. We see here, in fact, the prostate cancer. These are the prostatic glands. And the normal glands are here with this green basal layer, while in orange you see a tumor marker. So you can literally see how the tumor here invades the normal area. And this has been developed with OncoTyrol, a precision medicine or precision cancer medicine center in the western part of Austria. Important is that we not only generate beautiful images, but extract numerical data. And here you see, this is the normal area, as a normal prostate. On the x-axis is the basal cell marker, on the y-axis is the tumor marker. In a normal sample, you have here the population of prostatic basal cells on the glands here, and there's no tumor marker. But in the tumor area, here this population disappears and the tumor marker appears. So by using this, you can identify tumor areas based on quantification. The artificial intelligence is obviously something all of you are dealing with, and which revolutionizes currently our everyday life and also our professional work environment, and tissue agnostics is investing a lot in the development of AI-based solutions like classifiers to identify tissue. You see here skin, different compartments of a skin section classified by machine learning. Or here an lymphatic organ in a 6-color immunofluorescent staining. And what you do is that you just, when you run the training, you just label, like this is my germinal center, then you run the training, and what you get is then the automated quantification, the classification mask. And we, as I mentioned, we can also do this spatial biology, or what we call contextual tissue cytometry. You see the lines here, they show spatial relationships between cellular subcomponents. Here we have an example of mouse ovarian tissue where certain structures like primary follicles, secondary follicles, antral follicles have been automatically recognized. Or here endometrium, where glands, blood vessels, and stroma have been identified automatically by machine learning. Now, one of the maybe most intriguing technologies that we can offer at the moment is multispectral imaging with spectral unmixing. We have two systems here, one with an 8-slide loader, 8-slide stage, and one with a slide loader. And this is a very impressive reference paper from Harvard University in Boston, where they used our system to understand the immune response against the SARS-CoV-2 virus in thoracic lymph nodes of COVID-19 patients post-mortem. And please note when this was published, in August 2020, so just 5 months after WHO has declared the pandemic, at the time when there were still many governments who thought they don't need to do anything against corona, they just wait for herd immunity to come. And by using our TissueFax spectra system and our software, in a 7-color immunofluorescence protocol, I don't have the time to go into detail, but it's anyhow published. This group found that we will have a problem with long-term immunity in corona. I mean, nowadays we know it, but in 2020, this was really breakthrough research because the corona, as SARS-CoV-2, avoids the formation of follicles with germinal center, which has significant implications for expectations of herd immunity. And one of the maybe most intriguing and for me personally satisfying experiences comes from this project with the Ludwig Boltzmann Institute for Cancer Research in Vienna. And they found when you block the platelet-derived growth factor receptor, PDGFR, then you can effectively block the growth or the progression of a certain type of lymphoma. And this was an experimental treatment. And at the time, this was done on a single patient, a medical student, in fact, who was suffering from this type of lymphoma. And when they started to treat him, he had a remaining life expectancy of 6 months. So they treated him, and after 2 weeks, he was tumor-free. So that was really a revolution. And he's still alive today. So, and this was made possible because they could identify the effect by quantifying this with our system, TissueFax and the software. So, just to summarize, tissue image cytometry or TissueFax supports researchers to move from image to data by offering spatial analysis. It runs automatically, it is cell-based, it's cytometric or quantitative. It increases observer independence and gives improved reproducibility. I would like to acknowledge our academic collaborators around the world, in particular in Europe, but also Professor Batra in Australia, with whom I work on deep learning-based solutions, which we call the virtual histopathologist. So, with this, I would like to conclude my presentation. Thank you very much for your attention.